more bugs to become superbugs. And secondly, if you want to get the darn bug, you're going to have to be creative. You might have to use a cocktail of different drugs and get that thing by 2 different ways. That would be synergistic effects of the antibiotics. So we often use them in synergism. And this is a video actually for modes of action. I can't play the video, I'm sorry. I could if I took time, we could go back to the textbook thing and play it, but just, it's the same figure as the one above. Here's your cell wall. This is something you're going to have to understand for your test. How the peptidoglycan is structured. Remember I said it's a net-like molecule? You have alternating sugars. NAG, look in your textbook, stands for N-acetylglucosamine. Has anyone heard of glucosamine? Mm-hmm. Yes. What do you take glucosamine for?
Joints.
Your joints, your cartilage. So if peptidoglycan consists largely of NAG, which is like cartilage, that gives you an idea of what peptidoglycan is like. It's strong, but it's kind of flexible and tough like cartilage would be. And the NAG subunits alternate with another sugar called NAM, which is N-acetyluramic acid. So you have alternating units of sugars. NAG, NAM, NAG, NAM. And then notice the cross links between the NAM units. Those are called tetrapeptide cross links. So the enzyme is called a transpeptidase. You need a transpeptidase enzyme to make the cross links, which are tetrapeptides, in the peptidoglycan molecule. It's all very technical. I don't really expect you to memorize all of that. But it is easy enough to understand if you just take a minute. But what's going to happen? In penicillin and other drugs, like methicillin, cephalosporins, perhaps even vancomycin, bacitracin, they have this particular part of the molecule that's called a beta-lactam ring. That's the important part of the antibiotic. That's the part that makes it work. That's the part that binds with the active site on the transpeptidase enzyme, so the enzyme can't do its job to link up the cross links needed to build peptidoglycan. So this is how beta-lactam antibiotics interfere with peptidoglycan manufacture. They competitively inhibit the enzyme, the transpeptidase enzyme. Okay, they're showing you here, because you don't have good cross links, it weakens the wall so the cell could burst from osmotic pressure. Another thing that could happen is it can't make the cross plate because it can't build it. Can't build good peptidoglycan. It's going to be a weak wall. Can't build it fast enough, can't build it strong enough. So that's the mode of action for penicillin. Let's talk about spectrum of action. So this is ribosomes. How targets the ribosomes, you know that. Cytoplasmic membrane, you know that. Mechanisms of action, inhibition of metabolic pathways. Remember, we talked about anti-metabolites, like sulfa drug. And we also talked about heavy metals. Heavy metals will inactivate enzymes. So they're also considered anti-metabolites, because if you don't have an enzyme, you can't run your metabolic reaction. Okay, here's your sulfonamides. See, it's plugging up the active site so the PABA can't fit in. There's just all so much to cover in one semester, it's ridiculous. These are base analogs. So remember, the bases are A, G, C, T. Base analogs, it's just got a different letter. It's a phony letter. Oops. And if you've got phony letters, when you replicate your DNA, it's gonna have a lot of phony letters in it, and the cell's not gonna know how to read it or what to do with it. We'll talk more about this when we talk about viruses. But there are enzymes that we can use to inhibit the manufacture of DNA or the manufacture of RNA. So if we give the patient a drug that causes their bacteria or their viruses not to be able to replicate their DNA or make RNA, that's how we hurt them. Summary: 2 ways to interfere with nucleic acid synthesis. Use drugs that act as base analogs, so you have imposter building blocks, or interfere with the enzymes needed for DNA replication or RNA manufacture, or RNA synthesis. Prevention of virus attachment, that's about viruses. Okay. I'm looking for a spectrum of action, but I'm not seeing it here. It'll come up. Okay. Antimicrobial drugs. What do you want? All the good stuff. And here's spectrum of action. So antimicrobial drug, you want to be able to get it. You don't want it to be too expensive. You want it to be chemically stable so it can last on the shelf and also persist in the body and get to the site of infection. You want to be able to administer it relatively easy. Give my patient a shot or take it orally. If it's too hard to administer it, what good is it? You don't want it to be toxic or allergenic to your patient, and you want it to be selectively toxic so your patient doesn't get sick. But theoretically, we want to be able to get a lot of buzz with it too. So there's different things you would want to consider when you prescribe an antimicrobial. If your patient has a mixed infection, multiple microbes in there, you want a broad spectrum antibiotic. Maybe your patient has one kind of bacteria, a very specific kind. Why would I give you a broad spectrum antibiotic that's probably going to hurt your normal flora too, if I can give you an antibiotic that's selectively toxic, will specifically target just that one species of bacterium? So that would be narrow spectrum. Broad spectrum covers a wide range of pathogens, including gram-negative and gram-positive. Narrow spectrum specifically targets one particular kind of bacteria. There's 2 ways to go, depending on what's going on in your patient. If you don't really know what the infection is in your patient, but you got to do something, you might give them a broad spectrum. Or if you know your patient has a mixed infection, it's a UTI with gram-negative and gram-positive, give them a broad spectrum. If he's got something like tuberculosis, you're going to target that specific organism. That would be narrow spectrum. So that's the spectrum of action of the drug. Narrow and broad spectrum. Yeah.
Just to, like, I guess, illustrate that fact, I had a patient who had Klebsiella and MRSA at the same time. So we would do IV Vanco for gram-positive and IV Rocephin for gram-negative.
Right.
Treat them concurrently.
Yeah, good, because the Klebsiella is gram-negative and the MRSA is gram-positive. And the problem is there when people got a mixed infection like that, and he's probably immunocompromised, which is why he got a mixed infection in the first place. Really hard for the body to deal with all that at the same time. And so and actually what they did was, that was pretty targeted. That was actually a pretty specific treatment that they gave. Narrow spectrum Rocephin for the gram-negative and narrow spectrum Vanco for the MRSA. But you know, a lot of his normal flora is getting killed there too, which makes his immune system weaker even. You know, so you really got to count on the antibiotic to get those infections because his immune system's not going to be a lot of help. You know. Spectrum of activity. So here they're showing you, for example, isoniazid will cover mycobacteria and some gram-negatives. So that's how that figure works. Broad spectrum, a good example of a broad spectrum drug would be tetracycline. Tetracycline is our classic broad spectrum antibiotic. Tetracycline targets ribosomes. Gram-negative cells have ribosomes, gram-positive cells have ribosomes, even chlamydia and rickettsia have ribosomes. So it's your classic broad spectrum because it targets ribosomes that all the bacteria have. Problem, I have ribosomes in my cells too, so I might have some side effects with tetracycline, but it's a good broad spectrum drug. Everybody seems totally bored today. Are you okay?
No.
Yeah.
Well, the weather is helping.
Sam's having trouble even keeping awake. He had a difficult experiment this morning. He had to wait forever for his auger to melt. This is what your plates are gonna look like when you come back on Thursday, and you're gonna measure the zone of inhibition, which is all the way across. Can you see the little cursor all the way across? So you're gonna turn your plate upside down. You're gonna hold your ruler up to the bottom of the plate, so you don't have to open it, just turn it upside down, and you'll see those clear zones around the discs. What's happening here? Zone of inhibition, no bacteria growing, right? What's happening here?
Nothing.
Nothing. What does that mean?
The antibiotic's not working.
The antibiotic's not working. These bacteria are laughing in its face. They're having lunch on it. Right? They're growing right up and onto the disc. You can't see them growing on the disc because you're looking at the underside of the plate. But if you were looking at the top, you would see the bacteria growing right on the disc. The bacteria are completely resistant to the antibiotic in that disc. What if we compare this antibiotic in this disc to this antibiotic in this disc? Which one has a stronger effect?
Oh, the bigger one.
The bigger one? You naturally would think the bigger one, and you're probably right. However, antibiotics are chemical molecules. Some are larger than others. Some will diffuse into the agar faster than others. So these might actually be the same effectiveness. It's just that this one diffuses into the agar faster, so you get a bigger ring. So they actually have charts and tables, and you'll see it on the antibiotic chart for you to use at the bottom. There's like a scale where you can gauge. For this particular drug, a zone of inhibition this many